Hi everyone, this week in 1124 we're going to be going over PCR and DNA extraction, things that are pretty fundamental to pursuing a career in biology, so should be pretty cool. In lab this week, you're, as usual, going to have to read the lab background and describe the procedures. You'll be watching a video and a simulation. And then your lab summary this week will be a bit more straightforward. It's just answering a couple of questions, filling out a table, and then responding to a prompt regarding this semester's final, which I'll go over at the end of this presentation. But before we get into that, we're gonna go over today exploring why some people taste a substance that others don't. Typically, this is a very protocol-heavy lab, and this is a lot of skills that you'll probably be needing in the future if you go into biology. Even if you go into the medical profession, this kind of technique is very widely used in this kind of research. So, some background information. Everyone has this gene, TAS2R38. Apparently it can also be called Taster38, but not everybody has the same version. And so this is referred to as alleles, where everybody has this gene, but some people have different versions of that same gene. This gene produces taste receptors for the bitter taste. In general, the two versions that we're going to talk about are the taster version and the non-taster version. PTC is the chemical that triggers this receptor. And so you can think of this kind of like when we talked about neurotransmitters, the neurotransmitter would go and it would bind to the postsynaptic cell and then it would trigger a response. So PTC in this instance is kind of like that neurotransmitter, but it's found in food. So that chemical is going to go from the food, bind to those receptors on your tongue, and then send signals to your brain saying that you are tasting this bitter substance. And that is if you have the taster gene. So we're going to compare a person's ability to taste PTC with their version of the gene and see if we can figure out what that looks like. So typically we would be collecting a sample of cheek cells by scraping with a swab, extracting the DNA, copying the DNA, and then identifying the DNA. One, because we're virtual, we can't do this, and two, because of COVID, not a good idea to do this in a lab anyway. So we're going to be doing this through some simulations, and we will be providing you data, not this week. You'll be doing this separate lab summary, and not next week because we don't have labs next week. But in 2 weeks, you'll be getting data, and you'll be required to kind of analyze that data and figure out what's going on. So when you do PCR, it's completed in very tiny tubes. You would normally be, using these. You'd have to cut them into the appropriate number of strips and everything. This is lab protocols, but like I said, this technique is really ubiquitous. It's used almost everywhere. So I would definitely, if you plan on going into a biological career, look into these techniques. You would, in PCR, you're mixing very small quantities of clear liquids together, and then you have to make sure it works. So you have to be very organized. You need to make sure that the tubes are clean because DNA contamination is a problem. You don't want clay sticking to these kinds of things. And then you'll be placing the tubes into a heat block. And so the way that this works, you'll get into it, but it changes the heat to split apart the DNA, and then changes the heat again to let the polymerase work, and then changes the heat again so it binds back together. So you'll get into more detail in these simulations, and you'll be required to think about how the process works and everything. But this is kind of the nitty-gritty of how the lab materials would work. But for your purposes this week, all you have to do is complete the simulation, and then we'll work through what the DNA results mean next time we meet for lab. So, like I said, in your lab summary this week, you'll be thinking about your final. Of course, it's online because our whole section is online. But there are two parts to the lab summary, or this final lab summary. It's an Excel graphing activity and then a visual lab summary. So the visual lab summary will be over a previous investigation on which you've received the lowest grade. So you'll kind of get a chance to go back, see what you did wrong, and then think through the materials and try and get a much better understanding of what went on. So the three that you'll have to choose from, well, not choose, the three that you'll be looking at what your lowest grade is the yeast fermentation lab, the photosynthesis lab, and the Daphnia heart rate lab. Once you figure out which lab you'll be doing for your final visual lab summary, you will be putting together this report in a more visual fashion rather than just a written thing. You'll be making kind of a poster. So we'll provide you this template. Your main finding goes right in the middle. And this really needs to be 1 to 2 sentences about what the main takeaway of this experiment was. And so I'm not talking about what you yourself learned, but rather what the experiment tells you biologically. You'll have the side areas will have some text, and you'll still be talking your way through it, but basically the point of the visual lab summary is kind of imagine that you're at a conference and someone comes up to you and wants to hear about your experiment, and you're going to talk your way through the experiment, kind of an elevator pitch of what your science was. You'll be using FlipGrid to create a 3-minute presentation. Shannon actually went through and checked out a lot of the capabilities of FlipGrid. You can show your screen, but if you're working with limited technology, you're going to be able to be creative. You can print out your poster, your visual lab summary, and just like film it. You can you're gonna have to get creative, and so there'll be some like style points, but mostly the content is what you're going to be graded on. But by now you guys have been using FlipGrid for the whole semester, so hopefully you'll be able to get a better handling of it by the time the final rolls around. So as I mentioned, this is just kind of an overview of what you need to do. Watch the videos in simulation. Think about your final, and you'll be required to answer a couple of questions in the lab summary, which is posted on Canvas. And so as usual, if you have any questions, feel free to email me or pop into office hours.